Hello, my name is Maureen, and today I will be talking about the inheritance of traits. In this video, I will introduce asexual and sexual reproduction, pedigrees, and punnett squares. Throughout the video, you will learn new vocabulary in order to understand the subject in further detail. I will also use real-life examples to reinforce what you learned throughout the video with punnett squares and pedigrees. The first thing I will talk about are the two different types of reproduction, asexual and sexual. In asexual reproduction, the organism reproduces on its own. Its offspring will have the same DNA. Some examples may be bacteria and plants. However, not all plants reproduce asexually. Some bacteria go through a process called binary fission. In binary fission, they split into two, and so forth. Another type of asexual reproduction is budding. This means they bud an identical offspring right off themselves. Our main focus of this presentation will be based on sexual reproduction. Sexual reproduction occurs when two gametes get together to make a new organism. Gametes are reproductive cells. Some organisms that sexually reproduce are many animals, many plants, some protists, and some fungi. When talking about animals, the gametes are typically a sperm cell for a male organism and an egg cell for a female organism. Together, the gametes create a zygote, which is a fertilized egg. These organisms can be genetically diverse, and the offsprings are not clones. This occurs because gametes themselves often are different due to independent assortment and crossing over when they are made. Gametes contain thread-like structures called chromosomes. They usually have half the number of chromosomes than a somatic cell. The image under the asexual column shows binary fission, and the image below the sexual column shows gametes that are usually found in animals. Chromosomes contain genetic information that controls traits. Each cell in an offspring contains chromosomes from both parents. These chromosomes exist in pairs. In human body cells, there is a total of 46 chromosomes. Humans each pass on 23 pairs of chromosomes to the organism being created. 22 of these pairs are autosome, and the other pair are sex chromosomes that determine your gender, like seen in the picture above. Each chromosome also contains a gene. Each gene has genetic information for one trait. The different forms of a gene are called alleles. How the trait appears or is seen is called a phenotype. One example of a phenotype is your eye color. Phenotypes are controlled by two alleles called a genotype. This is not visible, but it can be estimated. Genotypes can be represented by uppercase letters, meaning it is dominant. When an allele is dominant, its trait will always show up no matter what the other allele is. Genotypes can also be represented by lowercase letters, meaning the trait is recessive. Recessive means that the trait the allele is carrying will usually not show up unless there are two recessive alleles. If a dominant allele is present, it is written first in the genotype. Last, genotypes can be either homozygous or heterozygous. A genotype that is homozygous means it has two of the same alleles. For example, uppercase H, and lowercase h are homozygous genotypes. A genotype that is heterozygous has two different alleles. For example, a genotype which has capital H and lowercase h is a heterozygous genotype. Like mentioned previously, traits are passed down by generations. Therefore, biologists created pedigrees. A pedigree is like a family tree. It shows the inherited traits that have been passed on from different generations. It can also help determine genotypes. The name comes from the French word pedigrees, that means crane foot. According to Bonnet, this comes from a visual analogy between the trace of the bird's foot and the three lines used in the English official registers to show the ramifications of a genealogical tree. The circles represent females and the squares represent males. The Roman numerals on the side represent the number of the generation, and the line between both parents is known as the marriage line. The lines below connect parents to children. Sometimes the pedigree may have a shaded shape, representing the trait being tracked. In this example, three shapes are half shaded because they have a heterozygous gene. The non-shaded shapes have the homozygous dominant gene. If there was a shape that was fully shaded, it would mean that it would have the homozygous recessive trait. In order to predict the trait that future offsprings may have, we use punnett squares. A punnett square is a square diagram that is used to predict the genotypes and phenotypes of a particular cross or breeding experiment. It is named after Reginald C. Punnett, who devised the approach. A punnett square can only be used for sexual reproduction. We want to know the probability of the organism having green leaves. On the punnett square, we see different alleles. In this case, uppercase Y represents yellow leaves, and lowercase y represents green leaves. One genotype from one parent gets placed at the top of the square, and the genotype of the other parent gets placed to the left of the square. It doesn't matter whether the mother or father gets placed at the top. Inside of each square, you're going to write down the symbol for the allele coming from the top and the symbol for the allele coming from the left. Remember that the dominant allele is always written first. When analyzing our punnett square, we make ratios. Our graph makes the ratio 0 homozygous dominant to 2 heterozygous to 2 homozygous recessive. This can also be converted into a percentage. Each square has a value of 25%. In this case, there is a 50% chance of the plant having yellow leaves because there are two squares with the dominant gene big Y, and 50% chance of the plant having green leaves because there are two squares with the recessive gene little y. Now, I will give an example using a human family. This is Lily and her parents. One day, Lily wondered why she had certain traits, such as her height, hair color, and eye color, so she decided to do some research. The first thing Lily decided to investigate was her height. Your height can be determined by your genes and the environment. First, she made a punnett square. In this case, we used the letter big H for tall and the little h for short as alleles. She and her parents are all tall, so they all need at least one dominant allele represented by a capital letter H. That is why her parents must have the genes big H or big H little h. When making the punnett square, she decided to put her mom as big H and her dad as big H little h. When she made the punnett square, she came up with the conclusion that there is most likely 100% chance that all of their children will be tall, since all of the results have the dominant gene big H. The ratio is also 1 homozygous dominant to 3 heterozygous to 0 homozygous recessive. Lily also made a pedigree. In Lily's case, she's tracking if anyone in her family is short or will be short. However, since no one is short, none of the shapes are shaded. Her pedigree shows that both of her parents have the dominant gene H, meaning that they are tall and they pass it on to their only child. Next, Lily decided to investigate about her hair color. Her dad, before his hair got slightly gray, had brown hair. Her mother, on the other hand, is blonde. She wanted to know why she wasn't blonde like her mother. When she made the punnett square, she made the alleles big B and little b. Big B is for blonde hair and the little b is for brown hair. She chose the genotype big B little b for her mother because if it were big B, Lily herself would have had blonde hair. Her dad, on the other hand, got the genotype b because he has a recessive gene which is brown hair. When she finished her Punnett square, she noticed that there was a 50% chance she would have gotten blonde hair, 50% chance she would have gotten brown hair. This also means that if her parents would have 4 children, most likely 2 of them would be blonde and the other 2 would be brunettes. The ratio is 0 homozygous dominant to 2 heterozygous to 2 homozygous recessive. In order to investigate a little deeper, she also made a pedigree. Her pedigree shows that both her and her dad had the recessive gene b, and therefore they would only pass the brown hair gene to the following generations. That is why their shapes are shaded. Last, she wanted to know why both her parents have blue eyes and why she is the only one with brown eyes. She chose the alleles big E and little e. Big E is for blue eyes and little e is for brown eyes. When she designed her Punnett square, she determined that both of her parents have to have the heterozygous genotype in order for her to have gotten brown eyes. The Punnett square showed that there was a 75% chance of her getting blue eyes and a 25% chance of her having brown eyes like she does now. Another way to interpret this is that if her parents would have had 4 kids, most likely 3 of the 4 would have had blue eyes and only one child would have had brown eyes. The genotype ratio is 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Lastly, for further detail, she also made a pedigree for eye color. This showed that both her parents have the dominant gene of blue eyes, and she ended up with the recessive gene. Therefore, she's the only shape that is shaded. Thank you so much for watching, and I hope you learned something new. Bye!